Good morning. It's April 27th and this is your daily brief in science. Here's everything you need to know. A new approach to gene editing delivery targets the producer cells, not just the virus-like particles, to improve efficiency and safety. A Whitehead Institute study published in Nature Communications shows that engineering producer cells can boost particle production and cargo loading across multiple editors and delivery systems. Researchers conducted a genome-wide knockout screen in human producer cells to identify gene functions that promote or hinder VLP production, linking each knocked-out gene to the particle via a barcode system. In the screen, nearly every human gene was knocked out, and disruptions were tracked in the produced particles to determine impact on production. Disabling a standout gene that normally limits guide RNA output increased functional cargo and boosted particle potency across several gene editing platforms. A negative regulator of particle production was identified. Its knockout raised guide RNA output and yielded more potent VLP cargo across tools and designs. Some knockouts increased production of particle components but reduced delivery potency, showing context-dependent effects and the need to balance pathway components for efficacy. There are nuanced gene effects where certain knockouts boost particle production but diminish overall delivery potency, signaling a trade-off to optimize protein cargo-limited settings. Another set of genes boosted particle protein production but reduced delivery potency. In protein cargo-limited contexts, these could still enhance potency. Collaborations aim to improve delivery to immune cells, neurons, and other cell types essential for treating various diseases. A new peer-reviewed study reviewing 177 studies warns that exposure to endocrine-disrupting chemicals like microplastics, bisphenol, phthalates, and PFAS, together with climate-related stressors such as heat and hypoxia, likely produces additive or synergistic effects that reduce fertility across humans, wildlife, and invertebrates. Endocrine disruptors can interfere with hormones and reproductive development across species, with examples including phthalates affecting sperm in animals and humans, and PFAS impacting sperm quality and development. These chemicals, found in many consumer products, are linked to hormone disruption and reduced reproductive capacity seen across species, including invertebrates, wildlife, and humans. The article notes more overlapping data are needed to understand the extent and mechanisms of combined effects, but the authors call for urgent measures to mitigate both chemical exposure and climate risks. Although each stressor has been studied separately, there is limited research on their combined effects. When they occur together, the effects could be additive or more harmful. Experts quoted in the article emphasize that simultaneous exposure to multiple stressors is likely to worsen fertility outcomes beyond either factor alone. The findings align with broader concerns about a global move toward lower fertility, supported by data from the University of Washington and earlier sperm count trends in Western countries. The findings bolster calls for policy action to reduce exposure to toxic chemicals and address climate change, pointing to regulatory successes like the Stockholm Convention's reductions in DDT and PCBs as examples of effective intervention. The study analyzed 177 studies and highlighted insights from researchers Suzanne Brander and Shanna Swan, noting the potential for greater fertility declines when combining toxins with climate impacts. The authors suggest these combined stressors may contribute to a broad global decline in fertility, building on prior findings that sperm counts and fertility are already decreasing in many populations. Researchers stress that joint effects are understudied and potentially more severe, prompting calls for policy actions to reduce chemical emissions and use, echoing past successes in reducing DDT and PCBs. The study underscores the ubiquity of endocrine-disrupting chemicals in consumer products and their cross-species effects, highlighting the need for integrated environmental health policies. A new study from researchers in Copenhagen and at Nanyang Technological University upends the idea that DNA is the first signal in a sunburn. Instead, RNA damage drives the skin's acute response to UV exposure, initiating inflammation and cell death in both mouse and human skin cells before DNA damage is even considered. In the study, removing the ZAK gene in mice stops the usual UV-induced inflammation and cell death, highlighting ZAK-alpha as a central mediator that translates RNA damage into the skin's sunburn response. The findings, published in Molecular Cell, could prompt revisions to textbooks and guide future research on UV-related skin damage. The research shows that RNA damage activates the ribotoxic stress response via ZAK-alpha, triggering rapid cellular reactions to UV exposure, while DNA damage is not the initial trigger in this model. This suggests a paradigm shift in how sunburn is understood and may influence approaches to prevention and treatment of UV-induced skin damage and inflammatory conditions. In both mouse and human cells, the early RNA-based response appears to set the stage for subsequent effects, with DNA damage following but not driving the initial response. The work emphasizes a specific molecular pathway that translates ultraviolet stress into an immediate skin reaction, separate from the traditional emphasis on DNA lesions. While the study focuses on acute responses, experts say the findings could inform broader research on skin inflammation and aging related to sun exposure. Overall, the study positions RNA damage and the ZAK-alpha mediated ribotoxic stress response as the primary spark for the brisk sunburn reaction observed after UV exposure. A new effort from UGA collaborators is building on Clemson's progress in transforming cotton and coloring fibers with eco-friendly methods, aiming to reinvent the crop itself through gene editing, synthetic biology, and advanced breeding. Led by Chris Saski, the project seeks to embed desirable traits directly into the cotton plant rather than adding them in later textile processing. If successful, the work could bolster South Carolina's cotton economy and place the state at the forefront of sustainable textile innovation globally. Clemson officials frame the project as advancing a land-grant mission of education, research, and service with potential benefits for growers, manufacturers, and consumers through expanded fiber value and versatility. The approach moves traits into the biology of cotton, potentially reducing resource use and environmental impact by shifting changes into the plant rather than the processing chain. This effort follows Clemson's prior advances in cotton transformation and eco-friendly fiber coloring, now extended through a collaboration with University of Georgia researchers. In 2024, Clemson University received an $11 million grant from the Bezos Earth Fund to pursue sustainable cotton research, underscoring the funding emphasis on environmental sustainability in textiles. The project is positioned to explore how gene editing and synthetic biology can create cotton with enhanced properties while maintaining profitability for farmers. The collaboration emphasizes a practical path from lab to field, aiming to deliver real-world benefits for growers, manufacturers, and consumers without detailing specific trait targets in this summary. The research highlights how embedding traits at the plant level could alter the production chain and fiber performance on a global scale. A fossil from Richard Spur in Oklahoma is reshaping our view of ancient reptile breathing. 3 specimens of Captorinus agouti show a 3-dimensional preservation of skin with an accordion-like texture, and researchers use neutron-computed tomography to map the skin wrapping and scaly pattern. This discovery, housed at the Royal Ontario Museum, reveals an early rib-based breathing system in amniotes, dating to about 289 million years ago. The team identifies a segmented cartilaginous sternum, sternal ribs, and intermediate ribs, with connections tying the rib cage to the shoulder girdle, supporting a coastal breathing mechanism. The fossils preserve soft tissues, including skin, cartilage, and proteins, pushing the preservation window for soft tissues back by nearly 100 million years. Protein remnants in bone, cartilage, and skin highlight ancient biomolecules that were previously unknown in fossils. The finding suggests rib-based ventilation may be the ancestral breathing mode for reptiles, birds, and mammals, which could explain a shift toward more active lifestyles and terrestrial diversification in amniotes. Researchers from institutions including Harvard and the University of Toronto are involved, expanding our understanding of early reptile evolution and land life. The specimens will remain available for ongoing study, keeping the ROM as a key resource for future work. These insights place Captorinus agouti at the center of a broader picture of how breathing strategies shape the evolution of land-dwelling vertebrates. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.